Welcome to the Clinical Education Initiative podcast, Conversations with CEI, where we feature conversations with clinical experts, their experience and insights on current health issues in the areas of HIV, primary care and prevention, sexual health, hepatitis C, and drug user health.
Hello, everyone, and welcome back to the CEI HIV Primary Care and Prevention podcast. I'm Tony Urbina, Medical Director of CEI's HIV Primary Care and Prevention Center of Excellence, Professor of Medicine at the Icahn School of Medicine at Mount Sinai, and your guide through the latest in HIV medicine. We're recording in May 2026, and it's been a genuinely remarkable few weeks for HIV cure research. Two major studies broke in quick succession. A Norwegian man who achieved long-term HIV remission after a stem cell transplant from his brother without having received a traditional CCR5 targeted therapy, and a small but striking phase 1 trial testing CAR T cell therapy, or chimeric antigen receptor T cell therapy, called CAR T, as a strategy to control HIV without antiretroviral therapy. Two very different biological approaches and two real signals. Today, we're covering both stories and putting them in clinical context. By the end of this episode, you'll walk away with hopefully three things: a clear picture of what happened with the Oslo patient and why it matters, even if it's not scalable, and a solid grasp of how CAR T is being adopted for HIV and what the early data actually show. And lastly, a practical framework for talking with your patients about cure research without overclaiming, overreaching. So let's get into it. Let's start with the Oslo patient. This case was published in Nature Microbiology in April 2026, and it's the kind of report that makes you stop mid-scroll. Here's the setup. A 63-year-old Norwegian man had been living with well-controlled HIV since 2006 on standard antiretroviral therapy, undetectable, no issues. Then he was diagnosed with acute myeloid leukemia and needed a stem cell transplant. His brother was identified as a match. And then on the day of the transplant, the oncology team made a discovery that changed the significance of the whole procedure. The brother was homozygous for the CCR5 delta 32 mutation, meaning both copies of his CCR5 gene were non-functional. If you're not immediately familiar with why that matters, here's the short version. CCR5 is the co-receptor that most strains of HIV use to enter CD4 T cells. People who are homozygous for the delta 32 variant, both copies deleted, are essentially resistant to HIV infection. Their cells don't express a functional CCR5, so the virus can't get in. The transplant proceeded primarily to treat the leukemia, but the medical team monitored his HIV status carefully. And over time, even after antiretroviral therapy was discontinued, viral suppression held. As of the publication date, he has been in HIV remission for more than 5 years. The Oslo patient joins a very short but growing list: the Berlin patient, the London patient, the City of Hope patient, the Dusseldorf patient, all of whom achieved long-term HIV remission after stem cell transplants from CCR5 delta 32 donors. Now, the clinical reality check, and this is important for when your patients read the headlines. Allogenic stem cell transplantation is a high-risk procedure. The conditioning regimens are cytotoxic. Graft versus host disease is a real and serious risk. The CCR5 delta 32 mutation is rare in the general population, somewhere around 1% for homozygosity in Northern European populations, much lower globally. Finding a sibling who is both an HLA match and carries this mutation is extraordinarily uncommon. So we are absolutely not recommending stem cell transplants for HIV. What this case does is validate the biology. It confirms again that targeting the CCR5 pathway is a legitimate and reproducible route to functional cure, and it tells us we need scalable strategies to get there. Which brings us to the second story. On May 11th, Reuters reported on early results from a phase 1 clinical trial testing CAR T cell therapy, chimeric antigen receptor T cell therapy, as a treatment strategy for HIV. If you work in oncology or follow it at all, you've heard of CAR T. It's been transformative for certain blood cancers, B-cell lymphomas, multiple myeloma, ALL. The basic idea is that you take a patient's own T cells, you engineer them in the lab to express an artificial receptor, the chimeric antigen receptor, that targets a specific protein on cancer cells. You infuse them back, and those cells become precision weapons against the target. The results in oncology have been really remarkable. The HIV field has been asking for years, can we adapt this for HIV? The challenge is that HIV is not a cancer. It's a virus that hides in lately infected resting CD4 T cells, not displaying much on the surface. But research have developed CAR T constructs that target HIV envelope proteins expressed on infected cells or that use CD4-based recognition. The trial used one such approach. The phase 1 data included 3 patients. 2 of the 3 maintained viral suppression after antiretroviral therapy was discontinued. Now that's preliminary. It's 3 people. It's phase 1, but those are the numbers and they are encouraging. The safety profile also appeared more favorable than what's seen with oncology CAR T. Cytokine release syndrome, the major toxicity concern, was observed, but at a lower severity in this setting. And that's clinically significant because it suggests the therapeutic window may be more manageable. The open questions here are real. How long do the engineered cells persist? Does the effect hold at 1 year, 2 years? Which patients are most likely to respond, and can we identify predictors? What does manufacturing costs look like at scale? So none of these questions are answered yet. This is not coming to your clinic next year. But the concept has cleared the first hurdle. It shows biological activity against HIV in vivo in humans with an acceptable safety signal. And that really matters. So let's pull back and put these 2 stories together because I think they're telling us something important in combination. Both the Oslo patient and the CAR T trial are targeting the same fundamental problem, the HIV reservoir, the latently infected cells that persist even on antiretroviral therapy and make a sterilizing cure so hard to achieve. The transplant approach replaces the host immune cells wholesale, and the incoming cells are resistant to HIV. The CAR T approach engineers immune cells to actively hunt and eliminate infected ones. Different strategies, same target. So for your patients who are following this news, and some of them definitely will be, I'd suggest a few framing points. First, acknowledge the excitement. These are genuine scientific advances, and they're worth talking about. Second, be clear about the timeline. These are early stage proof of concept studies. Cure is a long game research agenda. And third, if a patient is interested in clinical trial participation, direct them to Clinicaltrials.gov. Enrollment in cure-directed trials is how this research moves forward, and some of your patients may qualify. We are in a genuinely accelerating moment in HIV cure science. More trials, more converging biological strategies, more clarity on what the reservoir actually is and how to target it. And that's real progress. And it's worth communicating with both honesty and optimism. I'm Tony Urbina. Thank you for listening. We'll be back soon with more updates from the field. In the meantime, stay engaged, stay current, and take care of yourselves and your patients.
Thank you for tuning in. Join us next time for a new episode of Conversations with CEI. Visit us at CEItraining.org and follow us on CEI social media platforms.